In this video, we're going to cover G protein coupled receptors and second messengers. We're going to break down the structure of GPCRs and G proteins and how the binding of an agonist drug leads to the activation of that G protein. Then we'll explore what second messengers are, looking at the Phospholipase C inositol phosphate messenger system, as well as the second messengers cyclic AMP and cyclic GMP. And we'll also go through examples of drugs that interfere with these processes. So let's start this lecture by looking at the structural features of GPCRs and G proteins. Alright, if you've seen the previous lecture on the types of receptors, we introduced G protein coupled receptors, which is one of our main types of receptor superfamily, and they are found on the cell surface. They are cell membrane receptors. They're formed by proteins that cross the plasma membrane. So here's the structural representation of a GPCR. We have the lipid bilayer, which outlines the cell's exterior and interior regions. This is just a general model, okay? GPCRs are a diverse group found within our bodies. In fact, our human genome encodes for numerous G protein coupled receptors, totaling in the hundreds. They are formed from a single polypeptide chain, a single protein, typically comprising around 350 to 400 amino acids. And that polypeptide chain folds back on itself to cross the membrane 7 times in total. It has 7 transmembrane alpha helices, an extracellular N-terminal domain of varying length, and an intracellular C-terminal domain. GPCRs are sometimes referred to as 7 transmembrane spanning receptors, okay? These 7 alpha helices, which adopt a hydrophobic nature upon folding, enables the receptor to embed itself within the membrane, okay? And they are divided into 3 main classes, A, B, and C. They still share the same 7 transmembrane helix structure, but differ in other areas, such as the length of the extracellular N-terminus and the location of the agonist binding domain. Now, the N-terminal of this chain is situated outside the cell, and the C-terminus or intracellular domain contains the binding site for G proteins, okay? Whereas the ligand binding domain for these GPCRs is on the outside of the cell or deep in the cleft between the alpha helical segments within the membrane. As we covered in the previous lecture, the activation of a receptor depends on the binding of a ligand, specifically an agonist drug, to it. And the location where the ligand binds on a GPCR depends on the receptor type and the chemical makeup of the ligand itself. Small molecules like acetylcholine or monoamine transmitters like adrenaline can activate GPCRs. You'll frequently discover that the binding site is inside the receptor's pocket, which the alpha helices spanning the membrane have formed. On the other hand, for bigger molecules, for instance, a peptide molecule, the binding site is typically located within extracellular regions outside of the receptor. So essentially, where the ligand binding site is, where it's located, depends on the receptor and the ligand that activates it. Okay, so that's the structure of GPCRs. Let's now go through the structure of G proteins, because GPCRs are unable to signal or function without G proteins. But what exactly are G proteins? They are a large family of proteins, each encoded by distinct genes. Later in this lecture, we'll delve deeper into the various types of G proteins and their roles. So they have both protein and lipid components. The lipid part allows them to anchor to the inside of the plasma membrane. They are anchored to the inside of the membrane through attached lipid residues. So here we have the G protein complex, which consists of the alpha and beta gamma subunits. This is sometimes referred to as a heterotrimeric G proteins. Trimeric meaning that there are 3 subunits, and heterotrimeric meaning that the 3 subunits are different from each other. We'll come back to these subunits in a minute. Functionally, G proteins operate as molecular switches. They respond to the activation of G protein coupled receptors and effectively relay signals from the receptor to various effector systems and signaling mechanisms within the cell. This intricate relay mechanism is really important for communicating information from the receptor to the network of cellular response pathways. All right? And the reason why it's called G proteins is because they interact with guanine nucleotides. They interact with GTP and GDP specifically. And this interaction determines the activation state of the G protein. So when the G protein is interacting with the GDP, that G protein is in an inactive state and the signal is off. And when GTP is bound, it flips the G protein into an active or signaling state and the switch is on. Okay, so it's a molecular switch. And this fundamental mechanism demonstrates the significance of G proteins as molecular switches that are either activated or deactivated by the binding of GTP and GDP. Now going back to the subunits, alpha, beta, and gamma, the alpha subunit here is the one that contains the binding site for the GTP or GDP. And we'll consider the beta and gamma subunits as one complex rather than separate subunits, but you'll see what happens, okay? At rest, these subunits will exist as a trimer. They are bound together and anchored to the inside of the membrane. And GDP is bound to the alpha subunit. Remember, when GDP is bound, the G protein is in the off state. It's not signaling, okay? The molecular switch is off. So now let's subtract complexity and go through how we activate these gorgeous proteins. Exactly how do we turn them on? All right, so right now, the GPCR and G proteins are at rest. And remember, when the G protein is at rest, what happens? The nucleotide that is bound to the alpha subunit is what? GDP. The alpha subunit is snuggled up with GDP, this cutie right here. Now, when an agonist drug comes in and binds to the receptor, it's going to flip the G protein coupled receptor into its active conformation. Think of the agonist drug as a star, a star guest at a party. It's the one that shakes hands with the GPCR and wakes it up. And once it wakes up, it's going to create a high affinity binding site for the G protein. So then the G protein complex here is going to come in and binds to the activated receptor, okay? Then we get to the beautiful part. What's the beautiful part? When the GPCR and the G protein interact with each other, that causes the GDP molecule to dissociate from the alpha subunit. It causes the GDP molecule to say, see you later, to the alpha subunit, and GTP comes in, okay, jumps in, says hello, and binds to the alpha subunit. Now remember, it's the binding of the GTP that switches the G protein into its active form. This GTP is like the magic switch. When it comes on, it's going to transform the G protein into its active state. Okay, so the inactive form is bound to GDP, which is what we had at rest, and it's the binding of the GTP that switches the G protein into the active form. Okay, now when the G protein is activated, it's going to dissociate into two separate parts, two teams. First, we've got the alpha GTP team. That's the alpha subunit snuggling with GTP. Then there's the beta gamma team. So think beta gamma, okay, BY complex, bye-bye. It's going to say bye to the alpha subunit. The alpha GTP team separates from the GPCR, okay, and what happens then? Well, it can then move through the membrane and interact with its specific target protein, which is called the effector protein. And there are so many different effector proteins that are targets for activated G proteins. It can be an enzyme or an ion channel, okay? It really depends on the different types of G proteins that are being activated, as well as the cell types that the GPCRs and G proteins are located in. And the beta-gamma complex here can also do the same thing, okay? So it can also interact with an effector protein, which may be an ion channel or a kinase, let's say. Okay, so that's how we turn on the GPCR and G protein. That's a beautiful mechanism that sets in motion the activation of a G protein and its target protein or effector, okay, in response to agonist binding at the GPCR. Okay, so if that's how we turn it on, the question now is, how do we turn it off? Okay, because if there's a mechanism for turning things on, there should be a mechanism for turning things off. Let's go through it. So we said that when the alpha subunit is paired up with GTP, it's going to form the active alpha GTP complex, and it's going to interact with its effector protein. Here's the interesting part. This is pretty cool. Listen up. The alpha subunit possesses GTPase activity, okay? It's a built-in ability that allows it to act enzymatically and convert GTP into GDP. It can hydrolyze GTP to GDP. When this hydrolysis occurs, the alpha subunit is now coupled with a GDP molecule, and we release a phosphate group here. And this transition marks the shift from GTP-bound to GDP-bound alpha subunits. And remember, the GDP-bound subunit is the inactive state of that G protein, okay? So when GDP is bound, it's off, it's not going to be signaling. All right, that's pretty cool, right? But what does that mean? Well, it's going to leave the effector molecule and it's going to reunite with the beta-gamma complex, okay? Recall the binding of GTP to the alpha subunit initially prompted the separation of the alpha and beta-gamma subunits. Now, with the hydrolysis of GTP and the alpha subunit's association with GDP, the beta-gamma complex reconnects with the alpha GDP complex, okay? So it's going to be a family reunion. And this marks the completion of the cycle, bringing us back to the beginning where we had a GPCR and a G protein at rest. And they're going to be ready. They're ready to be activated by the binding of another agonist drug. Okay, so that's how the activation of GPCRs leads to the activation of G proteins. We're now going to go through the next step in this signaling cascade, which is what happens after a G protein is activated. This is where the concept of second messenger systems comes in. We're going to look at the role of second messengers in intracellular signaling, and then we'll go through some important second messenger systems. Okay? First of all, what exactly is a second messenger? So, a second messenger is an intracellular molecule that carries critical, very important information within a cell. So visualize it like this. Second messengers serve as intermediaries transporting or transferring important information from the receptor to the effector organ. Okay? So right here, we can see a GPCR that is being activated by an agonist drug. And when that receptor has been activated, it's going to activate a G protein, just like we covered before. Now what happens then? That activated G protein can then go on and interact with a particular target, such as an enzyme, which then produces intracellular signaling molecules that pass on information to other areas of the cell, okay? And this is where the term second messenger comes in. It comes from their position in this chain. So the initial messenger is the ligand, it can be a neurotransmitter or a hormone, okay? This ligand is known as the first messenger and is frequently the carrier of an external signal. The subsequent messengers, which are produced inside the cell, continue to connect the receptor and the target effector protein. So we refer to them as second messengers. And these second messengers are very different, and they can change a lot about how cells work. They will affect things like muscle contraction, cell division, cell differentiation, and metabolic processes, okay? And one of the most important things to know about second messenger systems is that they can integrate and amplify signals. When we talk about integration, we mean that more than one receptor, or more specifically, more than one GPCR, can have an effect on the same second messenger pathway. So this means that you can have different neurotransmitters and hormones and still have the same effect because they all work on the same second messenger pathways. And having these second messenger systems also lets us amplify the signals. And by amplification, we mean that the signal gets bigger as it moves through the chain. So if it stays in that agonist-bound activated state, more G proteins can come in, become activated, and then diffuse to their effectors. So that's one level of amplification. Another part of amplification is that when G proteins are turned on, they can turn on their effectors for different lengths of time. So the longer a G protein works with its effector, like an enzyme, the longer that enzyme is active and the more product is going to produce. Because of these second messenger cascades, we have different things that come into play that allows us for the amplification and integration of signals. All right? Okay. Now, earlier, we talked about how there are several different types of G proteins. Most of the differences between G proteins, I should say, functional differences, are due to differences in the alpha subunit. So this is how we usually group G proteins. And there are three important classes that you should be aware of, and they are the GS, GI, and GQ G proteins, okay? So these are named based on having different alpha subunits, and each of these different G proteins can activate different downstream targets and therefore activate different second messenger systems. Let's look at an overview before diving into key systems. So this is an example of two different kinds of GPCRs. We'll call them receptors 1 and 2, and they pair with a different kind of G protein. Because of this, the G protein will turn on a different kind of effector pathway. The final reaction of the cell will depend on which effector mechanism is turned on, okay? This right here is two of our most important second messenger systems. We have the phospholipase C inositol phosphate second messenger pathway, okay, which is on the right, and the adenylyl or adenylate cyclase second messenger pathway, which is on the left. Let's go through the PLC inositol phosphate pathway first. Okay, so we've already covered that there are multiple different types of these heterotrimeric G proteins. And for the most part, the main differences in these different G proteins are due to the different types of alpha subunits. And remember, we're focusing on three key players here, GQ, GI, and GS G proteins. And we're going to be starting with the second messenger system that is downstream of the GQ G protein. All right? When you get an activation, when you get activation of GPCRs that couple to and activate the GQ alpha subunit, you will end up with the activation of a second messenger system. It's like setting off a chain reaction leading to the activation of a second messenger system. This system is known as the phospholipase C inositol phosphate second messenger system, sometimes called the phosphorinositide second messenger system. That is a mouthful, okay? When this second messenger system is activated, we're going to have a couple of important outcomes within the cell. One outcome is the activation of protein kinase C. This is a key player in cellular functions, all right? And the second outcome is it leads to an increase in cytosolic calcium levels, which is really important for many processes. Calcium is a crucial intracellular mediator, and what calcium does depends on the cell type. For instance, in muscle cells, elevated cytoplasmic calcium triggers contraction, okay? While in cells responsible for secretion, such as those producing hormones, a surge in intracellular calcium sets off the secretion process. So cytoplasmic calcium is really important. It's a really important signaling molecule within the body. And we're going to break down how activation of GQ leads to the activation of this second messenger system, and of course, an elevation in calcium. All right. Now, we're going to use specific examples, okay? We're going to use a GPCR that couples to GQ, which is the M3 muscarinic receptor. And we're actually going to go through this a bit more when we get to autonomic pharmacology. So the M3 muscarinic receptor responds to the neurotransmitter acetylcholine. When triggered, it activates the GQG protein. You can find M3 muscarinic receptors in various locations throughout the body. These receptors play a pivotal role in smooth muscle, driving contraction upon activation. They're also found, like we mentioned before, in exocrine glands where their activation sets off secretion processes, okay? What we're going to do is, let's get rid of this text and zoom in. So when acetylcholine binds to and activates the M3 muscarinic receptor, it's going to activate the GQ alpha subunit of the G protein. And remember how we covered how receptor activation sets the G protein in motion earlier, okay? So once the G protein is activated, it can then move through the cell and interact with its target effector. And the primary effector protein for activated GQ is known as phospholipase C, and you'll often see this abbreviated as PLC. So when GQ is activated, PLC gets activated, this QD right here. Now, PLC, phospholipase C, is an enzyme that, as the name implies, cleaves lipids. It's an enzyme that loves lipid snacks, okay? So you find PLC in the membrane. It's bound to the membrane on the inside of the cell, right here. Now look at this. PLC is activated, right? It's going to cleave membrane phospholipids. And in particular, it cleaves a particular type of membrane phospholipid known as PIP2. One of its favorite traits is PIP2, which is short for phosphatidyl inositol 4,5-bisphosphate. Okay, that's a mouthful, so long to remember. So let's just call it PIP2 for simplicity. Okay, so when PLC cleaves this PIP2, it's going to form two products. All right, the first product that it forms is diacylglycerol, which is abbreviated as DAG. And the second product that is formed is inositol 1,4,5-triphosphate, okay, which is abbreviated as IP3. Now, I didn't include the chemical structure of PIP2, DAG, and IP3. What's important for you to know is that PIP2 is a membrane phospholipid that is cleaved by activated phospholipase, PLC, to form DAG and IP3, okay? So remember, PIP2 is a membrane phospholipid, and DAG, which is one of the products, is also membrane bound. However, the other product, IP3, is soluble. So when the PIP2 gets cleaved, the DAG stays in the membrane, okay? It's going to hang there, and the IP3 here gets released into the cytosol. Okay, so what happens now? Where do we go from here? We've produced DAG, and we've also produced IP3. So the DAG in the membrane then activates another protein. And the protein that it activates is an enzyme known as protein kinase C, or PKC. PKC is an enzyme that plays a versatile role with its effects varying depending on the type of cell. And this PKC, this enzyme, it can then phosphorylate a range of different proteins in the cell. And targets for PKC can include things like enzymes or transcription factors. Okay, we won't go through it in this lecture. But exactly what gets targeted will depend on the cell type where the PKC has been activated. All right, so the DAG is produced from the cleavage of PIP2, stays in the membrane and activates PKC, which can then phosphorylate other proteins. Now, shift your attention to IP3, okay, this little one here. What happens to this guy? Remember that I said that IP3 is water soluble. So when it gets released from PIP2, it moves to the cytosol. And one of the main targets of IP3 are the ligand-gated calcium channels on the membrane here of intracellular calcium stores. So IP3 opens these calcium channels. And as the channels swing open, stored calcium floods into the cytoplasm, okay? Now, what happens if you release calcium from organelles within the cell and into the cytoplasm? That's going to cause an increase in cytoplasmic calcium levels, okay, which was our second outcome that we said earlier. That increase in cytoplasmic calcium levels will then have a range of different effects. Again, depending on what sort of cell type is being activated. If it's being activated in a smooth muscle cell, for example, the increase in cytoplasmic calcium levels will lead to contraction of that smooth muscle. If it's occurring in exocrine cells, it will lead to secretion. So the effect depends very much on the cell type. All right, that's the PLC inositol phosphate pathway. Before we move on and talk about another messenger system, let's quickly summarize the entire pathway. In a nutshell, let's recap the key stages. It all begins with a GPCR receptor getting triggered. Okay, take acetylcholine for instance, which activates the M3 muscarinic receptor. These receptors link up with the GQG protein. When the GQ alpha subunit is activated, it moves within the cell, associating with its effector protein, known as phospholipase, PLC. This enzyme hangs out on the cell's membrane. And once PLC is activated, it takes a slice at the membrane's PIP2 lipids, producing two products. Do you remember the first product, diacylglycerol, DAG, which hangs in the membrane? Okay, it's going to activate protein kinase C, which is an enzyme. Okay, it gives it a tap on the shoulder, setting off a chain reaction that influences various cellular components based on the cell type. And secondly, we have IP3 here, which is water soluble. Okay, it's going to embark on a cytosolic adventure and interacts with ligand-gated calcium channels that are found on the membrane of intracellular stores. This example is the membrane of the endoplasmic reticulum, and the ligand-gated channels open, releasing calcium into the cytosol. Okay, so you're going to end up with an elevation in cytoplasmic calcium levels. That elevation in cytoplasmic calcium will then activate various different events within the cell, again, depending on the cell type. All right. So that's the PLC inositol phosphate system. Let's now talk about another important family of second messenger molecules, cyclic nucleotides. All right. What I'm going to do first is we're going to go through how the second messenger, cyclic AMP and cyclic GMP, are synthesized and broken down. We're also going to identify some key examples of drugs that interfere with these processes, and then we're going to go on and look at how GPCRs regulate cyclic nucleotides. Okay? So, second messenger molecules have three important components. First, they have a nuclear base, guanine for cyclic GMP and adenine for cyclic AMP. They then have a sugar. It's usually a ribose molecule. And to complete the trio, there's a phosphate atom that links it all up, forming a cyclic pattern that gives them their name. Okay, so the phosphate bonds to the sugar to form this sort of cyclic structural shape that you can see here. It's like a secret handshake. So these cyclic messengers have a nucleotide base, a sugar, and a phosphate that is connected to the sugar as well. These are two really important cyclic nucleotides for cell signaling purposes. And remember, when abbreviating their names, use a lowercase c followed by uppercase letters for the rest. Okay, this is just a convention that's used when describing these molecules. Let's explore how cyclic nucleotides are synthesized and what happens to them, starting with cyclic GMP. Look at the structure of cyclic GMP on the right here with its building blocks. We have a nucleotide base, a sugar, and a phosphate. Cyclic GMP forms from its precursor, guanosine triphosphate, GTP, thanks to an enzyme called guanylate cyclase. Once the cyclic GMP is formed, it can then be broken down to guanosine monophosphate. The enzyme that breaks down cyclic GMP is phosphodiesterase. So basically what happens with this breakdown is that the cyclic phosphate bond gets broken. Keep in mind that our body has different versions of phosphodiesterase, okay, each with specific tasks. For now, let's just remember the basics. Cyclic GMP starts from GTP and ends as guanosine monophosphates. And enzymes like guanylate cyclase and phosphodiesterase make it all happen. Okay, so phosphodiesterase, okay, the cyclic phosphate bond gets broken. So that's how cyclic GMP is formed. But exactly what is the role? What's its role within the body? One of its key jobs is regulating blood pressure and blood flow. When cyclic GMP is produced, it causes the relaxation of smooth muscle in our blood vessel walls, okay, or vascular smooth muscle. Remember, these blood vessels have muscle in their walls, okay? So when cyclic GMP is present, it eases that muscle, making the blood vessel wider. This widening is known as vasodilation, okay? Dilation, widening of the blood vessel. Vasodilation of a blood vessel leading to a specific organ boosts blood flow to that area. And this ability of cyclic GMP to cause vasodilation is incredible. It's something that we use clinically. There's a drug known as glycerol trinitrate, or GTN, which is useful as a treatment for coronary artery disease, angina, okay? The way that this drug works is that it donates a molecule called nitric oxide, okay? Nitric oxide is another signaling molecule. It's a gas, and what it does is it activates guanylate cyclase, the enzyme that produces cyclic GMP right here. When a patient takes GTN, the nitric oxide triggers guanylate cyclase, leading to more cyclic GMP production. This increased cyclic GMP brings about more vasodilation. Clinically, this means relaxed blood vessels reduce the heart's workload, helping it better match supply and demand, okay? This is why GTN is helpful in conditions like angina. Remember, cyclic GMP's role in widening blood vessels and aiding heart function. That's cyclic GMP. Let's move on to another important cyclic nucleotide, cyclic adenosine monophosphate, or cyclic AMP. Similar to cyclic GMP, cyclic AMP goes through a series of enzymatic steps to form and break down. Cyclic AMP is formed from a triphosphate nucleotide, ATP, which serves as the starting material right here. Adenylyl Cyclase, sometimes called Adenylyl Cyclase, is the enzyme responsible for converting ATP into cyclic AMP. Once formed, cyclic AMP is active. Okay, now there is a variety of phosphodiesterase that can also degrade and deactivate it, just like cyclic GMP. These enzymes break down cyclic AMP to form non-cyclic AMP, so just the non-cyclic monophosphate, the basic monophosphate. This process keeps cyclic AMP levels balanced and controlled. Okay, so comparing the formation of cyclic AMP to cyclic GMP, in both cases we start with the precursor molecule, ATP for cyclic AMP, which is converted into cyclic AMP by adenylyl cyclase, and similarly cyclic AMP is broken down into its basic form AMP by phosphodiesterases. All right. Moving on to its physiological role, let's focus on the heart as an example here. When cyclic AMP levels increase in cardiac muscle cells, the heart muscle contracts more forcefully. This means that in the heart, cyclic AMP enhances the force of cardiac contractions. Remember, cyclic AMP has various roles in the body, not just in the heart, okay? But this specific example demonstrates how various drugs can affect changes in cyclic AMP levels for therapeutic purposes. Absolutely beautiful. So in our earlier example, we saw a drug like glycerol trinitrate increase the production of cyclic GMP. Now, there are also other drugs that impact cyclic nucleotide levels by affecting their breakdown pathways, because earlier we saw GTN affecting the production of it. One such drug is milrinone. So milrinone targets a specific phosphodiesterase in the heart that usually breaks down cyclic AMP. When the patient is treated with milrinone, it inhibits this phosphodiesterase. Okay, so the breakdown of cyclic AMP is reduced. As a result, cyclic AMP levels increase in the heart. Since elevated cyclic AMP levels boost the force of cardiac contractions, milrinone acutely enhances cardiac output by increasing the amount of blood pumped out of the heart. Okay? Now, the thing is, cyclic AMP doesn't just affect one kind of cell. Okay? Even though we already talked about its role in the heart, we know that cyclic AMP interacts with different target molecules in different cells. Beautiful. One of the main things that cyclic AMP targets is an enzyme called protein kinase A. Protein kinase A, or pKa, has many different targets depending on the type of cell it's in. This variety is one of the reasons why cyclic AMP can have so many different roles or so many different effects in different situations. pKa, protein kinase A, works by phosphorylating other proteins, and it functions as part of a complex with 4 other molecules. Let's break this down. 2 of the parts of this complex are called regulatory subunits, while the other 2 are called catalytic subunits. When the catalytic subunits are linked to the regulatory subunits, they stay inactive because they don't have any kinase activity. When cyclic AMP comes into play, this all changes. So cyclic AMP binds to specific spots on the regulatory subunits, okay, these right here, causing them to separate from the catalytic subunits. By letting go of the active catalytic subunits, pKa is able to phosphorylate its different targets depending on the type of cell. And you'll see in a second, okay, why this is important, why I went through protein kinase A, okay? So that's cyclic nucleotides. Let's now focus on adenylyl cyclase and cyclic AMP and the role of GPCRs in regulating cyclic AMP formation. And let's look at some drugs that interfere with parts of this system, okay? So we have multiple different GPCRs in our body that exert their actions by regulating cyclic AMP levels. These GPCRs acting through cyclic AMP can be classified into 2 main groups: those that increase cyclic AMP production and those that decrease cyclic AMP production. Now, whether or not a G protein coupled receptor that regulates cyclic AMP levels increases or decreases cyclic AMP levels is dependent on the type of G protein that receptor couples to, okay? Remember, when we're talking about different types of G proteins, we're primarily talking about differences in the alpha subunit of that G protein. So remember the key players. We have GQ, GS, and GI. For this part of the lecture, there are 2 primary types. We have GS, stimulatory, and GI, inhibitory. It's in their names, okay? The GS protein stands for stimulatory, and when this gets activated, it increases the activity of adenylyl cyclase or adenylyl cyclase, its effector target, which leads to an increase in cyclic AMP levels, okay? So the GS protein interacts with the effector target, adenylyl cyclase, and stimulates the activity of the enzyme to increase cyclic AMP levels, okay? That's one type of G protein that regulates cyclic AMP levels, CAMP levels. The other type that regulates cyclic AMP levels is the GIG protein. This right here on the red. GI stands for inhibitory. Okay, so it's in the name. So when you get activation of a receptor that couples to GI, okay, I for inhibitory, the activated GI then interacts with adenylyl cyclase and inhibits the activity of that adenylyl cyclase. Okay, GI inhibitory, it inhibits the effector protein. If the activity of adenylyl cyclase is inhibited, what happens then? Well, you will have a decrease in cyclic AMP production. Okay, so you've got two different ways that different types of G proteins can regulate adenylyl cyclase and therefore cyclic AMP levels. So what this means is that GPCRs that couple to GS will have the opposite effects of GPCRs that couple to GI. Let's break this down further. Let's slow down for a second. Let's start with an example of a GPCR that couples with GS. The example we're going to use is the beta-1 adrenergic receptor. So the beta-1 adrenergic receptor is a GPCR, a G protein coupled receptor. And one of the main places that you find the beta-1 receptor in the body is in cardiac muscle cells. The agonist for these receptors, or one of the agonists for these receptors, is adrenaline. Okay? Adrenaline, which is an important neurotransmitter released during the fight or flight response by the sympathetic nervous system, acts as one of the agonists for these beta-1 receptors. When adrenaline binds to and activates the beta-1 adrenergic receptor, that receptor then recruits and activates its G protein. And the G protein that the beta-1 receptor is coupled to is the GSG protein. Keep in mind that GS stands for stimulatory G protein. Okay, so activation of GS leads to an increase in adenylyl cyclase activity. And this heightened activity in turn raises the level of cyclic AMP. All right. Now remember earlier, we talked about how one of the main targets for cyclic AMP is protein kinase A. As cyclic AMP levels rise, protein kinase A gets activated to a greater extent. There's an increase in the activation of protein kinase A. So you can see it right here. Now in cardiac muscle cells, protein kinase A serves to phosphorylate voltage-gated calcium channels. And by phosphorylating, okay, adding phosphate groups, these particular types of channels, it actually increases the rate at which these channels open. Now what happens when we increase the rate at which these channels open? By increasing the rate of calcium channel opening, you end up with more calcium moving into the cytosol of those cardiac muscle cells. So remember what we talked about earlier in this lecture. When you have an increase in calcium levels in the cytoplasm of muscle cells, that increase in calcium levels increases the rate and force of cardiac contraction. So this is the mechanism by which adrenaline acting at the beta-1 adrenergic receptor increases heart rate and force of contraction. It's through the activation of the GSG protein, which then increases adenylyl cyclase activity to increase cyclic AMP levels. Absolutely incredible. Okay, so that's an example of a GPCR that couples to GS to increase cyclic AMP levels. Don't you just love it when everything starts to connect? Now, let's look at the opposite of that. Let's now look at a GPCR that couples to the GIG protein to decrease cyclic AMP levels. To illustrate this, we're going to look at the M2 muscarinic receptor, another example in which the neurotransmitter acetylcholine is the agonist. This receptor is also found in cardiac muscle cells, but activating this M2 muscarinic receptor has the opposite effect on the heart as activating the beta-1 adrenergic receptor. When acetylcholine binds to and activates the M2 muscarinic receptor, that leads to the activation of the GIG protein. Remember, GI stands for inhibitory. It inhibits adenylyl cyclase. Okay? So that activated GI protein then moves to the membrane to interact with adenylyl cyclase, and it reduces the activity of the adenylyl cyclase enzyme, which then leads to a reduction in what? In cyclic AMP levels. Okay? What happens then? What happens when there's a reduction in cyclic AMP in the cardiac muscle cells? Well, as we touched on earlier, decreased cyclic AMP levels translate to reduced calcium influx into the cardiac muscle cell, okay? This reaction occurs for the same reasons we explained in the previous example. Higher cyclic AMP levels correlate with an increased frequency of calcium channel openings. So if you have a reduction in cyclic AMP levels, you're going to reduce the opening of those calcium channels. So what's our outcome here? What's going to happen? If you're reducing the opening of the calcium channels, you have less calcium coming into the cytosol, and you're going to have less contraction, all right? Okay. Another downstream effect of activating these M2 receptors is an increase in the permeability or conductance of the cell to potassium. This change leads to more potassium moving out of the cell, inducing hyperpolarization. So the rate of cell depolarization and contraction slows down, effectively reducing the heart rate. Right? In summary, GPCRs that couple with the GI protein, such as the M2 muscarinic receptor, work to decrease cyclic AMP levels, which then affect calcium influx and potassium permeability, ultimately leading to a reduction in heart rate. So this is showcasing how GPCRs, when aligned with the GI protein, regulate cyclic AMP levels to influence cellular functions and overall body responses. Before we end this lecture, let's quickly summarize the various ways that different types of GPCRs and G proteins impact cyclic AMP levels. Okay, so on the left side here, we see a GPCR coupled with a stimulatory G protein, GS. We use the beta-1 adrenergic receptor as an example. When activated by adrenaline, the alpha subunit of GS triggers adenylyl cyclase, okay, this enzyme, increasing cyclic AMP levels. This leads to enhanced cellular responses through the activation of protein kinase A, all right? And on the right-hand side, we see the effects of inhibitory G protein activation, GI, okay? Here, the M2 muscarinic receptor activated by acetylcholine couples with the GI. The alpha subunit of GI dampens adenylyl cyclase activity, leading to decreased cyclic AMP production, which then decreases pKa activation and its downstream targets. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video, or you can view the entire playlist.